Okay, question two. So essentially, if we look at it in its entirety, a bit of a longer stimulus in terms of the information. So then essentially, I just want to go through it, try and understand it as I go through, and just make some notes throughout it. So, scientists have investigated how the nucleus accumulates the enzymes that it needs to function. Okay, they found a possible answer while studying a particular mutant form of the SV40 virus. So we're looking at mutant and normal, I assume. A mutant polypeptide produced by this virus was not able to get into the host cell's nucleus in the way that the normal was meant to. So it's mutant versus normal and how they actually get into the host cell. So I'm just making these connections as I go. The polypeptides for the normal form and the mutant form is shown below. A sequence of 7 amino acids from the middle of the polypeptides were shown. So when I look at this, I'm thinking, okay, what's similar, what's different between them? This is the normal, this is the mutant. We look at section B, we look across, and these two are clearly different, okay? So we know that there's going to be some sort of substitution that's occurred in terms of the mutation. So as I actually digress with the question, both the normal and mutant forms of the polypeptides were cut into 3 smaller chains, A, B, C, and A prime, B prime, and C prime. Each polypeptide was put into a cell and then its accumulation in the nucleus was measured. So you're measuring the accumulation of it in the nucleus. And so what we've actually gained here is you've got A, B, C, A prime, A, B prime, C prime, and then the normal and the mutant. And the two things that stand out to me at the moment are that N accumulates in the nucleus, so the normal one, and B does as well, but the rest of them don't. Okay. So based on the results in the table, scientists proposed the hypothesis that the sequence of 7 amino acids, pro, etc., enabled the polypeptide to cross the nuclear membrane and accumulate in the nucleus. What evidence supports this hypothesis? So if we come back and knowing that sequence is important, I will come to this data. So what this data shows me is that the normal strain or the normal polypeptide actually accumulates in the nucleus. And that is largely due to the presence of B, because when B was put in, that polypeptide did accumulate in the nucleus as well. So what that shows me is that it actually consists of N and B. And so they actually pass through the nuclear membrane, as I said, and accumulated. However, polypeptides without the sequence did not. So it's all about this comparison question. This works and this works. So I want to say, okay, first and foremost, the polypeptides N and B pass through the nuclear membrane and accumulate in the nucleus. That's my first part, because they did. And that's just observing what the data says. But when I look at the rest of these, they don't. None of them actually go through. And so that shows me then that other polypeptides without the sequence did not accumulate. And it's a very simple sort of comparison question, and people actually did really well on this, 52%. And I say really well, this is for a short answer of getting 2 out of 2 marks. 52% of the state got this correct. But the whole point and why I've underlined is however. It's to include however, whereas, or in contrast, just to show that you're comparing what actually occurred in terms of N and B with the others that did not actually accumulate in the nucleus. And it's all just about what you extract from the data. And what helped me throughout this, just to bring it to a close, is by highlighting these two things, but just noting throughout what was occurring. We had a normal, a mutant, and obviously the sequence of amino acids within the polypeptide chain B and B prime were different. So moving into part B. So scientists can make polypeptides with any sequence of amino acids. They also have techniques for inserting these polypeptides into the cytoplasm of cells. So they can have techniques of inserting into the cytoplasm. One scientist predicted it was the presence of lysine in the 3rd position of the amino acid sequence, which we identified before, that enabled the polypeptide. So I'm looking at the 3rd position of the sequence to accumulate in the nucleus. So what she did is she designed a controlled experiment to test her prediction. From the get-go, she used a large sample of identical cells, so she's already done that, and all cells were kept in the same environmental conditions, so as controlled variables. Outline the other steps that the scientist took to complete her experiment. So what a lot of students would have done, and they did do, was actually include these two statements within their response, and they obviously couldn't be awarded marks because that was already stated within the experimental design that she actually undertook. So if I'm outlining this, it's very, very simple. We look at what we're going to do. We want one that shows that these polypeptides went through and one that doesn't. So if we actually get one group of cells that are inserted with these polypeptides that contain lysine in position 3, we'll let them gather and we'll let them do that. But the second group, it would make sense for have amino acids with a different amino acid in position 3. So different group of cells and insert a different polypeptide sequence in there that does not contain lysine in position 3. That way you have a contrast occurring, so your idea of control. Then what you would want to do is accurately record which polypeptides accumulate in the nucleus, very similar to what occurred here. And obviously what we'd want to do here is, even though there's a large sample of identical cells and there's same environmental conditions, we need to ensure that we repeat the experiment, or we do multiple setups for that experiment to ensure that our results are concordant or consistent. I think that's the really big thing. So when you go through this, it's very simple. Getting two groups, one control, one actually experimental group, and then accurately recording what occurs, and then have that experiment being repeated. And I've obviously noted here, which I actually emphasized before, that the question has already noted those two variables. A large sample of identical cells were used and cells were kept under the same environmental conditions. But guys, 3 marks again. I've given 4 particular points, but again, it's the logic. This, then that, then that, then that, so that the assessor can actually see my logic and understand it.